My name is Dr. Amy Herford. I am a theoretical biologist. The big question that my research asks is, are populations increasing or decreasing in abundance? To illustrate this research question, I'm going to use the example of salmon lice or sea lice, and this is research that was mostly led by my recently graduated master's student, Matt Rittenhouse. Predicting the abundance of sea lice or salmon lice is challenging for three reasons. Firstly, the sea lice have different life stages. So the calamus, the pre-adult, and the adult stages are all stages where you have the sea lice attached to the salmon, like in the picture that you saw on the previous slide. But the nopali and the copepodid stage are planktonic stages. So in order to predict the abundance of adult sea lice, you need to think about what's the rate that eggs are produced by the adults, what's the rate that the eggs turn into nopali and then mature through each of the stages, and also what's the chance of dying in each of the stages. So it's more complicated than just considering one life stage. To complicate matters even further, the rate of maturing between different life stages depends on temperature, and that's what's shown in the graph on the top right-hand the top right-hand graph. And also the probability of dying in any of the life stages depends on salinity. And we know that temperature and salinity aren't constant throughout the year. This is the bottom graph. We know that temperature and salinity change throughout the year, and so somehow we have to take these three pieces of information and fit them together if we want to be able to predict whether sea lice populations are going to grow or not. This is my lab does mathematical modeling, and this is the mathematical model that my graduate student came up with. And so if you're wondering why as a biology major you're required to take Math 1000, on the left-hand side here we're seeing the rate of change in the number of planktonic or infectious or copepodid sea lice stages. This is a derivative with respect to time. We take that system of equations and turn it into some computer code and then solve that computer code, and we get our answer about whether the population of sea lice is increasing or not. And my lab is committed to open science, so this computer code was produced in the software R, which is free. Anyone can download it from the internet. The actual computer code that was part of this research is freely available from a website, Figshare. So after you've installed your version of R, you can then download the code and run it and actually make your own conclusions about whether sea lice are going to grow or not. And if you want to learn more about what the model does, the manuscript that describes this is freely available from a preprint server called bioRxiv. So here's one of the results. It was part of Matt's thesis. In the red region, after 5 years, the number of sea lice is less than 1, and so that's a population that's pretty much going to extinction. In the blue region, after 5 years, the abundance of sea lice is up around 100 or more, so this is a population that's growing, and we're seeing this as a function of the mean temperature and the mean salinity. So again, we know that temperature and salinity are varying throughout the year, but the mean is giving us a good indication of whether the population is going to grow or not. My lab doesn't just focus on sea lice. We're interested in a number of different questions, and these include trying to link host movement with the evolution of virulence. So this is how likely parasites are to kill their hosts. I'm also interested in the evolution of generalism in parasites. So this is how many hosts does a parasite infect. I'm interested in the evolution of molecular mimicry in parasites. So these are particular parasites that have protein sequences that are mimicking protein sequences that are found in the human body, like cells that are found in your heart. I'm interested in how different treatment strategies affect the level of antimicrobial resistance in hospitals. Models of optimal foraging, mutualism in bivalves, animal recolonization and spread, species responses to climate change, and selective logging in the Amazon. As you might guess, it's not really so much a specific system or species that I'm interested in, it's more the methodological approach. And I'm interested in research where developing mathematical models and analyzing mathematical models is an approach that's going to give you a lot of insight into the dynamics of what's happening with the population. If you're interested in learning more about this, I would really love to hear from you. My email is aherford@mun.ca. I also have an office in the Science Building right across from first year biology labs. You can visit my website to learn more about my research or read my blog. And in particular, if you're interested in mathematical biology, I'd recommend going to the Society for Mathematical Biology's website. I think there's a lot of really useful resources available from that page. Thanks for listening. Hope you are interested in learning more about math biology in the future.